Hello guys, this is Prime Concept. In this video, we want to look at a very important instrument. So we want to look at optical instruments. One of the optical instruments and the easiest we want to consider is the simple microscope. The simple microscope. The simple microscope. Microscope, which we have considered before anyway, but we just want to mention it again. Another name for your simple microscope is your magnifying glass, right? Or your hand lens. So it's either you call it simple microscope, or you call it magnifying glass, or you call it what? Hand lens. So in this video, I'm going to analyze how large can your simple microscope magnify an image, right? Yes. Now, a simple microscope is actually your convex lens that works under a certain condition. Remember when we started the concept of thin lenses, we looked at various positions where you can place your object and then where the images would be formed. And one of the positions we considered was when an object is placed between the focal point and the optical center of that lens. We saw that when you place an object in this particular position, that the image is going to be highly magnified and the image is going to be erect and virtual. So it is that condition that for which a convex lens can be used as a simple microscope. So what is a simple microscope? A simple microscope is a convex lens for which the object is placed between the focal point and the optical center. So that is the definition. I believe it's clear to you. A simple microscope is an optical device or instrument for which the object is located between the focal point and the optical center of the lens. That's a simple microscope. Now let's derive the formula that can be used to determine the magnification of a simple microscope. Now, this is our convex lens. This is my convex lens, like this. So let's still repeat the diagram. Now we know that this point is our focal point. This is our focal point. Now, remember this convex lens would behave as a microscope, this time a simple microscope, when the object is specifically placed between the focal point and the lens. This is when this would be true. Now, if we have our object O at this point and want to see where the image will be formed and the size, it means we're going to have a parallel ray so that when it hits the center of my lens, it comes through this point, right? Yes, it refracts through the focal point there. Now, if I consider one going through the optical center, so you would simply notice that these two rays are diverging from each other. They are separating, spreading away, meaning that they will not meet at any point on this side. So if I trace them backwards, I trace this backward like this, then trace this backwards like this. So you notice that both of them would meet somewhere on the same side as the object. So where they meet is where our image is formed. So this diagram we have is a diagrammatical representation for a simple microscope. So you can see that our image is erect. Our image is virtual because it is formed on the same side as our object. So our image is virtual, it is erect and highly what? Magnified. So you can see relative to your object, the image is very big, and that is why it is called a simple microscope. A simple microscope is a device that you use to view close objects so that you can see them in a more magnified way. So that is what we have like this. Now, what is the focal length? Remember this distance now from here to this point is going to now be our V, right? And then this distance from here to here is going to be our what? U. So what is the formula for magnification? We derived a relationship for magnification in one of my videos. that generally magnification for a lens is either given as m equals v divided by u, or we can say m is equal to what now? 1 minus v by f. So we derived this relationship, right? So the magnification is either this or this, right? Yes. Now, so that if since you can see that our image is formed on the same side as our object, it means that v is going to be what? Negative. Now, we want to consider a case, a special case, where this image is located at a point which we know as the least distance of distinct vision. The least distance of distinct vision is denoted with the symbol d. So if v is at that point, then it means our image distance is going to be negative d. Why am I using negative? Because my image is formed on the negative side. That's why I'm using negative, and it is virtual. So that's why we're using negative. So if we consider for least distinct vision, then our magnification m is going to simply be 1. If I put, okay, minus, my v now is minus d, all divided by f. So if I consider this, I would have that m is going to be 1 plus d over f. So this is what I have. So this is the formula for magnification of a simple microscope when the image is formed at the least distance of distinct vision. So I believe that this concept of simple microscope is clear. So do well to understand it, and also understand this diagram very well, because in my next video I'll be talking of the compound microscope. The compound microscope also forms within it a part of the simple microscope. So that's why this concept should be clear, so that the compound microscope would be even clearer to you. Thanks for watching this video. Please do well to hit the subscribe button, like, comment, and share with 10 of your friends. Tell them this is prime concept. Thank you.